Hello, my name is Michelle Ulima Barahokhma, and in this video, I'll be presenting my systematic review on how DNA damage response can enhance immunotherapy. Now, to understand this topic, we first need to understand what DNA damage response actually is. Our DNA is constantly under attack. Every single day, cells faces damage from both internal and external sources. Internally, the biggest threats come from reactive oxygen species, which are byproducts of our normal cellular metabolism that can chemically modify or break DNA strands. Externally, things like UV radiation, ionizing radiation, and even chemotherapy drugs can cause significant DNA damage. The type of damage matters a lot. You can have base mismatches, single strand breaks, or the most dangerous types, double strand breaks, where both strands of the DNA are simultaneously cut. Each damage has its own dedicated repair systems. The most critical repair system is homologous recombination or HR, which is mainly controlled by BRCA1 and BRCA2 proteins. When these proteins are mutated and no longer functional, the cell loses its most reliable repair tool, and it has to fall back on less accurate backup systems, one of which involves an enzyme called PARP. Now, here is where immunotherapy comes in. We currently have checkpoint inhibitors, drugs that blocks BT1 and PDL1, which essentially releases the brakes on the immune system so it can recognize and attack cancer. But the problem is that these only work for only 20 to 30% of patients, mainly those whose tumors already have a high mutational burden. The majority of patients, especially those what we call immune cold tumors, meaning tumors with little to no cell proliferation, simply do not respond. So the central question this review is trying to answer is, can DDR defects help us change that? Why this matters? This matters for three key reasons. First, immunotherapy is one of the most promising cancer treatments available today, but its reach is still very limited. Second, DDR defects, which are actually very common across multiple cancer cells, may be generating signals that the immune system can detect, essentially making tumors more visible and attackable. And third, if we can confirm and standardize this relationship, it changes how we select patients for treatment. Instead of only testing for PDL1 expression or tumor mutational burden, we could also be testing DDR status, which would open the door to immunotherapy for much larger groups of patients who are currently not eligible. The aim of this review was to examine DDR effects and enhance immunotherapy across different cancer types, with a specific focus on C-gasting pathway as the central mechanism. The review was aimed to critically assess the current evidence and identify the research gaps. For methodology, a systematic review was conducted across four databases: PubMed, NCBI, Google Scholar, and Science Direct, which cover studies from January 2016 to December 2025. The keywords used were DNA damage, C-gasting, immunotherapy, and PARP inhibitors. Around 85 articles were initially identified and was narrowed down to 17 full-text articles. 17 was included in the discussion and were mainly narrative reviews, with one also incorporating phase 2 clinical trial data. Main mechanism. Before getting into the PARP inhibitor specifically, it's important to map out the mechanism so everything that follows makes sense. The process works in three sequential steps. It starts with DDR defects. When cell can't properly repair DNA, damage accumulates and unrepaired DNA leaks out of the nucleus to the cytosol of the cell. This is significant because DNA normally should not be in the cytosol. The cell innate immune system recognizes this as a danger signal, what we call as DAP, or damage associated molecular pattern. This triggers the second step, activation of the C-gasting pathway. C-gas is an enzyme that detects cytosolic DNA and produces a signaling molecule called 2,3-C-GAP, which activates STING. STING then drives type 1 interferon production, a powerful immune alarm, while also activating NFKB, which produces a pro-inflammatory cytokine and chemokine like CXCL10 that recruits immune cells into the tumor. The third step is where PARP inhibitors comes in. They amplify the entire process in the tumors that already have HR deficiency, and that what makes them a much powerful tool in this context. The role of PARP inhibitors. So what exactly happens when PARP inhibitors is given to an HR deficient tumor cell? Normally, PARP enzyme repairs single-strand DNA breaks. When PARP is blocked, those breaks goes unrepaired and gets converted into double-strand breaks, especially when the DNA replication fork runs into them during cell division. In a normal cell, HR would and double-strand breaks fix these double-strand breaks. But in BRCA mutated HR deficient cell, there is no functional HR. The cell is left with a catastrophic unresolvable DNA damage. This is the concept of synthetic lethality. Two, two individual tolerable defects, PARP inhibition plus HR deficiency becomes lethal when combined. The cancer cells essentially destroys itself. But beyond ju- just killing the tumor cell directly, all the damaged DNA floods into the cytoplasm, activating C-gasting pathway. This triggers a type 1 interferon response, releasing pro-inflammatory cytokines like IFNY and TNF alpha, which activates NFKB, all of which creating an inflamed tumor microenvironment. The tumor cells also upregulate MHC class 1 molecules, meaning they get better at displaying their abnormal proteins, their antigens, to CD8 plus T cells. And interestingly, PARP inhibition also upregulates PD-L1 on tumor cells through the C-gasting and ATM-AGR pathway. This explains perfectly why combining PARP inhibitors with PD-L1 and PD-1 checkpoint blockade is such a rational strategy. The PARP inhibitors inflame the tumors and makes it visible, while the checkpoint inhibitors remove the brake that was stopping T cells from attacking. Finally, DNA fragments that leaks out of the tumor cells can also act on neighboring dendritic cells in what we call paracrine C-gasting response, further boosting antigen presentation, activating more CD8 plus T cells, recruiting NK cells, and ultimately driving immunogenic cell death, where the immune system fully eliminates the tumor. So, what did this reviewed study actually find? Across all 7 studies, there was consistent association between DDR defects and improved immunotherapy response, but the strength of that association varied depending on the cancer types. Breast cancer, particularly triple-negative breast cancer with BRCA1/2 mutation, showed clearest and most uniform benefit. This makes mechanistic sense because BRCA mutation directly impairs HR, creating an ideal condition for PARP inhibitor synthetic lethality and c-Cas activation. Studies showed that combining PARP inhibitors with PD-1 inhibition could significantly improve pathological complete response rate. Ovarian cancers also showed strong potential. High-grade serous ovarian cancer has one of the highest rates of HR deficiency, around 50%, making it an ideal candidate for PARP-based treatment. Maintenance PARP inhibition after platinum chemotherapy already extends progression-free survival, and the reviewed data suggests adding immunotherapy could further extend this benefit through immune memory. NSCLC, or non-small cell lung cancer, was more complicated. Unlike breast and ovarian cancers, where BRCA mutations are dominant, DDR defects, NCLC tends to have more heterogeneous DDR defects involving ATM, CHEK2, and other mediators. Studies the heterogeneity means immune signaling is less uniform and less predictable. Studies suggest that radiotherapy immunotherapy combinations may be more effective in NCLC, since radiation induced DNA damage in DDR compromised cells can amplify C-gasting activation similarly to PARP inhibitors. Overall, the most promising treatment strategy was DDR inhibition combined with PD-1 or PDL2, PDL1 blockade, but with the clear understanding that cancer types and patient selection matters enormously. As promising as all of this sound, it is important to be honest about the quality of the current evidence. Scientifically, the biological rationale is very strong, but when it comes to clinical translation, there are significant gaps. The biggest issue is study design. Most included studies are narrative reviews from early phase 1 and phase 2 clinical trials. Phase 1 are primarily designed to assess safety. They are not powered to definitively confirm whether or not a treatment works. Phase 2 provides clear efficacy signals, but with typically fewer than 50 patients, the statistical power for definitive conclusion is very limited. The second major issue is lack of standardization. Different studies use different PARP inhibitors, different checkpoint inhibitors, different treatment sequencing protocols, and different biomarker tests to define DDR deficiency. Without standardization, results can't be reliably compared across studies. So, the evidence strongly suggests the hypothesis that DDR detects can and immunotherapy works synchronously, but it is yet not sufficient to form a definitive clinical guideline. There are several important limitations worth addressing. First, follow-up periods across included studies average are under 18 months. In oncology, long-term data, 3 to 5 year survival outcomes, is essential before confidentially recommending a treatment strategy. Short follow-up means we can't properly assess whether responses are durable or whether resistance develops over time. Second, the vast majority of patients in these studies come from Western cohorts, Western academic centers in the US and Europe. This is a serious representation problem. Asian populations, including Southeast Asians, are significantly underrepresented. Despite carrying a disproportionately high burden of cancer, like nasopharyngeal carcinoma, which is especially prevalent in this region, crucially, DDR defects patterns in Asian populations also differ from Western populations. Western cohorts tends to show more BRCA1-2 mutation rates, while East Asian cohorts more commonly show ATM and CHEK2 defects. This means the findings may not directly translate to our population. Third, biomarker testing was not uniform across studies. HRD scores, BRCA sequencing, and functional assays all measure different aspects of HR deficiency and don't always agree. Until testing is standardized, patient selection will remain inconsistent and unreliable. Based on the evidence and several recommendations emerge, Based on the evidence, several key recommendations emerge. First, in clinical practice, DDR profiling, including HRD scores and ATM check-2 sequencing, should be implemented alongside existing PDL1 and TMB testing to give a more complete picture of which patients are likely to benefit from immunotherapy. Second, PARP plus PD1 and PDL1 combination trials should be prioritized with standardized protocols for drug selection, dosing, sequencing, so that the results can actually be compared and built upon. Third, and most urgently, large-scale phase 3 trials that include diverse global population, specifically Asian and African cohorts, are critically needed. Without this, any clinical guidelines developed will have a fundamental and generalizability problem. And finally, biomarker testing needs to be standardized. Functional assays that measures actual HR capacity appear to be most predictive and efforts should be made to validate and implement these in clinical settings worldwide. To conclude, DDR defects are not just a marker of cancer progression. They represent a genuine biological vulnerability that can be exploited to improve immunotherapy response. The C-gasting pathway is the central bridge between DNA damage and immune activation, converting immune cold cell tumors into inflamed, treatment-responsive environments. PARP inhibitors are currently the strongest tool for amplifying this effect, particularly in BRCA-mutated breast and ovarian cancer. However, this field is still in its early stages. Moving forward, DDR profiling has a real potential to become a foundational biomarker in precision oncology. expanding immunotherapy access far beyond the current 20 to 30% who benefits today. I'm Michelle. Thank you for watching my systematic review presentation.